Hi, my name is Alyssa Kell, and today I'll be teaching you about head anatomy. So we're going to be covering the nasal cavity, sinuses, oral cavity, pharynx, and the eye. So here you can see the nasal cavity. This is a lateral view, and you can see up top, you can see the olfactory nerve traveling across the ethmoid bone and down through the cribriform plate. And then we have three nasal concha. So you have the superior nasal concha, the middle nasal concha, and the inferior nasal concha. And these are three ridges of bone that increase the surface area of the nasal cavity, which that helps warm and moisturize air as much as possible before it reaches the lungs. And these concha are covered in mucosa. And then we have three nasal meatuses under each concha. So you have the superior nasal meatus, the middle nasal meatus, and the inferior nasal meatus. And these are just openings under each of the concha that connect the nasal cavity to the paranasal sinuses, which we'll cover on the next slide. And then finally, this is the nasal septal cartilage, which divides the tip of the nose into the right and left nostrils. And the nasal cartilage is just the tip of your nose that some people get pierced. And here we have a lateral view and an anterior view of the paranasal sinuses, which are just hollow spaces within the cranial bones. And these are also lined with mucosa to help humidify any inhaled air. And these sinuses open into the lateral walls of the nasal cavity through the nasal meatuses. So first, in this yellowish color, we have the frontal sinus, which is within the frontal bone, and it opens into the nasal cavity through the middle meatus. And then next, in green, we have the ethmoid sinus, which is within the ethmoid bone, and it opens into the nasal cavity through the superior and middle meatuses. And then in blue, we have the sphenoidal sinus, which you can only see on the lateral view because it's pretty deep. And this is anterior to the pituitary gland, which is within the sphenoid bone. And then finally, we have the maxillary sinus, which is a large sinus that opens into the nasal cavity through the middle meatus. And this is in the orange color. So now we're going to be looking at a view of the upper mouth or the palate, and we're going to go over the different kinds of teeth that are in the palate. So first in purple we have two incisors, which are the most medial teeth that are adapted for cutting. And then in green we have a canine tooth, which is pointy and longer. And then in blue we have two premolars, which are transitional teeth. They're between the canines and the molars. And then finally in pink we have three molars, which are large and flat teeth that are in the posterior mouth that are meant for grinding food during mastication. And then you have the same thing, so two incisors, one canine, two premolars, three molars on both sides of your upper mouth and your lower mouth. And next we're looking at the soft palate, which is formed by muscles in the posterior oral cavity, and they're all covered in mucosa. So first we have the uvula, which is a process that dangles down off the soft palate. And then the first arch that we're looking at is more anterior, and this is called the palatoglossal arch, which is formed by the palatoglossus muscle. And then the more posterior arch is called the palatopharyngeal arch, and this is formed by the palatopharyngeus muscle. And then next we're looking at the tongue. So the tongue is divided up into the posterior root and the anterior body, and these two areas are divided by the sulcus terminalis, which is a V-shaped groove that just divides the two areas. Okay, now we're looking at another view of the oral cavity, specifically the salivary glands, and then we'll look at the hypoglossal nerve. So there are three major salivary glands that secrete saliva, and saliva is important to keep the mouth moist and prevent tooth decay, and it also is essential to our sense of taste. So there are ducts that carry saliva from each of these glands to the mouth, The first gland is the sublingual gland, which is the smallest salivary gland that lies on the floor of the mouth, and the right and left sublingual glands combine to create a horseshoe-shaped mass inferior to the tongue. Next, we have the submandibular gland, which lies along the body of the mandible, and then finally the parotid gland, which is the largest salivary gland that's just anterior to the ear. Next, we're looking at the hypoglossal nerve, also known as cranial nerve 12, which enters the neck through the hypoglossal foramen on either side of foramen magnum and then travels under the tongue. And it provides somatic motor innervation to most of the tongue muscles, and this allows the tongue to change its shape and move, and this is essential for mastication and speech. And then next you can see the hyoid bone, which is a unique bone that doesn't articulate with any other bones, and you can see it in both of these images here. Okay, now we're looking at an image of an oral cavity. So you can see the two incisor teeth, the one canine, the two premolars, and three molars on their upper left mouth. And then you can see these arches and the uvula of the soft palate. So the anterior one is the palatoglossal arch, which then leads to the uvula, and then the posterior arch is the palatopharyngeal arch. Now we're looking at a view of the pharynx, and this is a sagittal cross-section. So the pharynx is a muscular tube that extends from the nasal and oral cavities to the larynx and esophagus, and it's divided into three parts. The most superior part is called the nasopharynx, which is posterior to the nasal cavity. The middle part is called the oropharynx, which is posterior to the oral cavity. And the inferior part is called the laryngopharynx, which is posterior to the larynx. And then after the laryngopharynx is the esophagus, which we'll get into in a second. And now we're looking at some muscles of the pharynx and the oral cavity. So on the left, you see a posterior view of the pharynx, and on the right side of that image, you can see the blood vessels and nerves. And then on the right, you have a sagittal cross-section of a head and neck. So first you can see the hyoid bone, which is that C-shaped bone that wraps around the neck. And here you can just see the posterior one side of it, and the other one would be over here behind all of these nerves and blood vessels. And then on the sagittal image, you can see the auditory tube opening, which is where the middle ear connects to the nasopharynx. And then here you can see the genioglossus muscle, which is a large muscle that attaches the tongue to the mandible. And this is the mandible right here. And this muscle acts to produce tongue depression and protrusion. And the mylohyoid muscle supports the floor of the mouth. It attaches to the mandible and the hyoid bone. Next, we have the pharyngeal constrictor muscles, which you can see from both the posterior view and the sagittal cross-section. You can just see the walls of the pharynx that they wrap around. These muscles attach the pharynx to the hyoid and larynx, and they squeeze food inferiorly from the pharynx into the esophagus, which then peristalsis takes over in the esophagus. These muscles receive somatic motor innervation from cranial nerve 10, or the vagus nerve, and they receive general sensory innervation from cranial nerve 9, or the glossopharyngeal nerve. And then on the picture on the left, you can see there are three separate pharyngeal constrictor muscles, the superior, middle, and inferior constrictors. Next, you can see the superior cervical ganglion of the sympathetic trunk, which is where any sympathetic nerve that's going up to the head synapses. And these nerves originate in the thoracic and lumbar levels of the spinal cord and then travel up the sympathetic trunk through the thoracic cavity. And then the ones that go to the head synapse here. Okay, now we're going to look at the anatomy of the eye. So the eyeball is made up of three layers. The sclera is the most superficial layer, and this is a tough protective layer. The choroid is the middle layer that contains a lot of blood vessels, and the retina is the deepest layer that contains the rods and cones that respond to vision. And then the ciliary body is a ring of smooth muscle that's continuous with the choroid layer, and the ciliary body acts to control the shape of the lens and depth of focus. And then the zonular fibers attach to the ciliary body, and when the ciliary body contracts, the zonular fibers pull on the lens to change its shape, which brings us to the lens, which is a transparent, flexible structure that changes shape depending on how far you're looking in order to focus light onto the retina. Next we have the cornea, which is a continuation of the sclera at the very front of your eye. And here we have the iris, which is another ring of smooth muscle that's continuous with the choroid layer. And this contains pigment, which makes up the colored part of the eye. And the iris covers part of the lens and controls the amount of light that's let into the eye. Okay, next we have the vitreous humor, which is a clear fluid that fills the posterior eyeball, and light must pass through the vitreous humor before it hits the retina. And next we have the optic nerve, or cranial nerve 2, which is continuous with the retina, and the optic nerve extends posteriorly out of the orbit to the brain and enters the skull through the optic canal of the sphenoid bone. And this only has special sensory function, which would be vision, so it relays information from the photoreceptor cells in the retina to the brain. And here you have the optic disc, which is a raised disc on the retina where it connects to the optic nerve, and there are no photoreceptor cells in the optic disc, so it doesn't sense light, and this is sometimes called the blind spot. So now I wanted to explain how binocular vision is made possible through the optic chiasm. So this image that you see on the right shows the visual field on the top, and the binocular visual field is what's visible by both eyes at the same time, while the right and left visual fields are only visible by one eye. And the visual field is when both eyes are wide open and looking straight ahead. So points A and D are peripheral points that are only visible in the monocular fields. So for example, if you look as far to your right as you can, this would be point D, and images from your right peripheral vision only hit the nasal retina of your right eye. So the nasal retina is just the medial part of the retina, and the temporal retina is the lateral part. Points B and C, as well as point FP, which stands for focal point or fixation point, these are all points that are in your binocular field, so you see them with both eyes at the same time. And then the bottom of this picture shows the eyes and then how the retina reaches back into the optic nerve, which then branches into the optic tracts. So the nasal retinal fibers cross at the optic chiasm, and this is what gives us binocular vision. And then the optic tracts are the combination of retinal and nasal fibers. So the left optic tract is formed from fibers from the left halves of both retinas, which would be the right nasal fibers and the left temporal fibers. Now we're going to go over a case of visual field defects. So visual field defects can be caused by many different neurological disorders, but they all involve a lesion to optic nerve fibers somewhere on the route from the orbit to the visual cortex in the occipital lobe. So in this picture, you can see the optic nerve starting in the posterior orbit, and then the nasal fibers cross at the optic chiasm, and then they branch back to the lateral geniculate nucleus in the optic tract. And then from the lateral geniculate nucleus, after they synapse, they'll branch out into the optic radiation fibers, which then reach the occipital lobe. So You can determine the location of the lesion based on what parts of the visual field are still processed. So the area of the visual field that's no longer seen is called a scotoma, or a blind spot. And this may be caused by glaucoma or even diabetes if there's a blocked blood vessel in the retina or optic nerve. So in case A, you can see that this person has no vision in their right eye, and this is due to a lesion at the right optic nerve before they cross at all. So that leads to the complete blindness. And then case B is called bitemporal hemianopia, and this is when there's damage to the optic chiasm, which is where the nasal fibers cross. So there's a loss of vision in the temporal or peripheral fields of both eyes. So this could be called tunnel vision. And then case C is called left homonymous hemianopia, and this is when there's damage to the right optic tract. So this contains nasal retinal fibers from the left eye and temporal retinal fibers from the right eye. So this person has lost their peripheral vision in the left eye and central vision in the right. Case D is called left superior quadrantinopia, and this is when there's damage to the right superior optic radiation. So there's a loss of vision in the left superior quadrants of both eyes. And then finally, case E is called left homonymous hemianopia with macular sparing. So this is similar to case C, but the macula lucia, which is part of the retina where light focuses, is conserved, so this person still has the central vision. And this might not be as noticeable because they can still see parts of each quadrant. And it's important to understand how the optic nerve fibers travel and cross on their way to the occipital lobe to know how an injury will affect their vision. And to test for a visual field defect, the ophthalmologist might do a perimetry test. So the patient will stare into a machine that has a bowl-shaped apparatus, and they stare at the center of the bowl as lights flash in different areas of their visual field. And the patient will push a button each time they see a flash of light, and wherever they don't see the flash of light is where they have vision loss. So finally, this structure in animal eyes is called the tapetum lucidum, which gives animals night vision. It increases the amount of light that passes through the retina. This isn't found in humans, but it's in various vertebrate animals. And you can see in the picture that it looks blue and metallic. And this is posterior to the retina.